Hello, my name is John Fleck, and this is my case connection number 2 presentation, where I will go over case number 1 and case number 4. So case number 1 details pyruvate kinase under 2 conditions, one in a normal cell and one in a cancerous cell. So the enzyme pyruvate kinase is an essential component of the glycolytic process. The purpose of this enzyme is to facilitate the reduction of phosphoenolpyruvate, or PEP, to pyruvate. So this is the final and irreversible step of glycolysis. It's also the rate-limiting step of glycolysis. In normal mammalian cells, there are several means by which pyruvate kinase is regulated. First and foremost, hormone control. So pyruvate kinase is part, again, of the glycolytic process, which is naturally anabolic. The primary anabolic hormone of the body is insulin. Insulin binds to tyrosine kinase receptors on the surface of cell membranes to activate the processes of glycolysis, glycogen synthesis, the TCA cycle, fatty acid synthesis, and protein synthesis. Conversely, pyruvate kinase is inactivated by the primary catabolic hormone of the body, which is glucagon. Glucagon stimulates the processes of glycogenolysis, gluconeogenesis, beta oxidation of free fatty acids, and lipolysis. Secondarily, pyruvate kinase is also regulated by specific molecules. Fructose 1,6-bisphosphate is a molecule that allosterically activates pyruvate kinase. Fructose 1,6-bisphosphate uses a feed-forward mechanism to increase flux through glycolysis and enhance production of pyruvate. When energy levels are sufficient, pyruvate kinase is inhibited by ATP. So again, the entire metabolic process to produce energy starts with glucose, which is then converted to pyruvate. Pyruvate is converted to acetyl-CoA. Acetyl-CoA is converted to NADH, which is pushed through the electron transport chain to produce ATP, which is the energy currency of the cell. So in situations where there is adequate ATP present, the flux through glycolysis would be inactivated, reducing energy production. So the amino acid alanine also regulates the activity of pyruvate kinase. Pyruvate kinase increases the levels of pyruvate, which may be transaminated in skeletal muscle to the amino acid alanine. Alanine is then transported back to the liver, where it is transaminated again back to pyruvate. Pyruvate is readily available in the liver when alanine levels are high, thereby inducing an inhibitory effect on the enzyme pyruvate kinase. So that was a regulation of pyruvate kinase in a normal cell. Cancerous cells have different biochemical properties that promote other metabolic processes. So pyruvate kinase M1 is common in normal cells and pushes substrate through, again, the glycolytic pathway toward eventual energy production in the electron transport chain. Pyruvate kinase M2, on the other hand, which is common in cancer cells, instead pushes substrate through the pentose phosphate pathway and also circumvents this TCA cycle, which leads to a production of lactate. This difference is important because the cancer cell's primary objective is cellular proliferation. To grow as quickly as possible, cancer cells utilize the capabilities of the pentose phosphate pathway to increase NADPH production, which helps to combat oxidative stress and also to produce excess ribonucleotides, which are essential for cell division. Cancer cells also uptake an enormous amount of glucose from the blood and convert it to lactate through anaerobic means rather than the standard metabolic processes used by healthy cells. This process is called the Warburg effect and causes cancer cells to retain vast sums of anabolic substrates in their microenvironments, which causes them to consume excess energy and proliferate at an abnormal rate. So that was case 1. Case 4 is a scenario where a 2-month-old female is presented with jittery spells after breastfeeding. So the mother reports that she's only been giving the baby breast milk since she was born, and that the baby has recently started sleeping for 2 hours between feedings, which she had not done previously. So blood tests were run, and the only significant finding was that the baby was hypoglycemic, so she has low blood glucose levels. Epinephrine then was administered in hopes of stimulating the glycogenolysis pathway and increasing glucose levels in the blood, but no increase was seen. Rather, there was an increase in blood lactate. Physical exam shows an enlarged liver 4 centimeters below the costal margin, which is indicative of hepatomegaly, or an enlarged liver. So again, blood glucose levels were able to be increased following a feeding, but they could not be sustained during a fasting period. So the child in this case likely has a glycogen storage disease. Specifically, she probably has developed a glucose 6-phosphatase deficiency, which is otherwise known as von Gerk's disease. This diagnosis can be made based on the child's inability to stabilize her glycogen stores in a fasting state, and despite an administration of epinephrine. Under normal circumstances, epinephrine activates the glycogenolysis through the alpha agonist pathway, as I mentioned. When glycogenolysis is activated, glycogen in the liver is broken down into glucose, where it can enter the bloodstream and be used by cells. Glycogen is a branched polymer of glucose. These branches allow for easy release of glucose 1-phosphate, which can readily be used again during the process of glycolysis. Because this child is unable to utilize her glycogen stores during the fasted state, the child's body cannot properly promote glycolysis and quickly becomes hypoglycemic without regular feedings. The hypoglycemic state explains the sudden emergence of the excessive sleep patterns exhibited by the child. It also explains the hepatomegaly. So while von Gerk's disease does interfere with glycogenolysis, it does not interfere with glycogen synthesis. The child's body is still able to store glycogen in the liver in anticipation of breaking it down later during a fast. Because glycogenolysis is inactivated, excess glycogen stores cause the liver to swell and produce the hepatomegaly that the child is experiencing. So the other differential diagnoses were possible based on symptoms, and they were very closely related to metabolism, but ultimately they were not possible for several reasons. So while a hereditary fructose intolerance would cause hypoglycemia, the child in this case has only been fed breast milk. So breast milk consists of only lactose and galactose. Fructose is only common in foods such as fruits, honey, and artificial sweeteners. So this child cannot have an intolerance to fructose because she hasn't consumed any. The child also cannot be experiencing galactosemia because she is exhibiting the wrong symptoms. Children with galactosemia typically present with jaundice, diarrhea, and vomiting in conjunction with the other symptoms that this child is experiencing, such as an enlarged liver and lack of energy. Also, a blood test would indicate high levels of galactose. Of course, from this scenario, the only significant finding was that she was hypoglycemic. Glucose 6-phosphate dehydrogenase is a regulatory enzyme of the pentose phosphate pathway. Under conditions where there is a deficiency in this enzyme, the pentose phosphate pathway cannot proceed as needed and results in a reduction in the concentration of NADPH in the blood. So as I mentioned in the last scenario, NADPH is required to combat oxidative stress. It does this by regulating levels of the antioxidant glutathione. When glutathione levels are reduced, Red blood cells will be under oxidative stress, which causes them to lyse and leads to cell death. So under these conditions, hemolytic anemia and jaundice will take place, neither of which this child is experiencing. Patients with glucose 6-phosphate dehydrogenase deficiency would present again with anemia and reduced NADPH levels in the blood. The physical exam would also show that the child would have an enlarged spleen and jaundice, neither of which this child was experiencing in this case. So while the other differential diagnoses were legitimate, they're reasonable, the child in this case likely has C, a glycogen storage disease or inability to use glycogen. Thank you.